Hello friends, today we are going to study about composition, structure and function of carbohydrates part 10. In this part, we'll study about the sugar code. What is the importance of carbohydrate in a biological system apart from its function of giving energy and as a structural component? It is found as a conjugate with proteins and lipids. So glycoprotein or glycolipids or glycosaminoglycans, in all these glycoconjugates, the importance of carbohydrate is the carbohydrate function as a sugar code. That means it is a code that store information. Okay, in the last video we already learned that carbohydrates help in the proper protein folding. Carbohydrate function as a tissue specific marker. In this video we'll learn more about what information carbohydrates convey to our body. So one branch of biochemistry, which is glycobiology. This branch is the study of structure and function of glycoconjugates. While study about glycoconjugates, their structure and function, various specific roles of these oligosaccharides or carbohydrates were noticed. The functions are: these carbohydrates or oligosaccharides, they help in intracellular targeting of protein. They also help in cell-cell interaction. They also function in tissue development and as well as an extracellular signal. For example, if we take any oligosaccharide which is attached with a protein. Now suppose this oligosaccharide consists of 14 monosaccharide units. 4 types of monosaccharides which are repeated in a particular way to form a 14 monosaccharides unit consisting of a oligosaccharide. Now the linkage can be different. It can be 1 to 2, 1 with 3, 1 with 4, 1 to 6, 2 to 3, etc. Now these carbohydrates can also be in the form of alpha or beta conformation. These carbohydrates can also be sulfated in nature. So that means this oligosaccharide is enormously rich in structural information. Varieties of oligosaccharide can be present with a protein or lipid. Every protein having different set of oligosaccharides, every lipid having different set of oligosaccharides. Same protein or lipid found in different tissue can have a different set of oligosaccharides which differ in their monosaccharide unit, which differ the number of times these monosaccharides are repeated, which can differ in their conformation alpha and beta, which can differ in the linkages they are present. Some monosaccharides will be sulfated in nature. So we can get a diverse, a wide range of different varieties of a oligosaccharide set which constitute a sugar code. So this carbohydrate or this oligosaccharide is enormously rich in structural information. Now, one such example is lectin. What are lectins? These are proteins that read sugar code and help in many biological processes. Lectins are the proteins that can read sugar code and can help in many biological processes. These lectins are found in all organisms. They bind with carbohydrate with high affinity and specificity. For example, these are the most common examples. In animal, the lectin protein is a mannose binding protein A. Definitely it will bind to mannose. In case of viral, influenza virus hemagglutinin is a lectin which bind to neuraminic acid, that is acetalic acid. Polyoma virus protein 1. It also bind to neuraminic acid, but see the linkage difference. It is 2 with 6, here 2 with 3. Rest all is same. In case of bacterial enterotoxin, the ligand for it is galactose. Cholera toxin, the ligand for it is GM1, which is a ganglioside. This cholera toxin, what it causes? It causes diarrhea. Why they cause diarrhea? Because of cholera toxin, there occur absorption of water in intestinal cell. So the toxin, the cholera toxin is a lectin. It binds to the target cell, that is intestinal cell, through the oligosaccharide present in GM1 ganglioside. Similarly, if we talk about mannose binding protein A, this mannose binding, mannose is attached to a newly synthesized protein which are meant to be present in lysosome. Where proteins are formed, we'll study during cell biology, but where proteins are formed, the ribosomes. Either in free ribosomes or rough endoplasmic reticulum. The ribosome attached with the rough endoplasmic reticulum. So after proteins are formed, they are passed to other cell organelles like Golgi complex. So mannose are attached to the proteins which are meant to function in lysosome. How the cell will know which protein it has to send to the lysosome? So for that some signal is attached, and that signal is this mannose, mannose 6 phosphate. The signal is mannose 6-phosphate. If any protein is having this signal mannose 6-phosphate, then the cell will know the destination point of this protein is lysosome. So it has to transport to the lysosome. So for this, if Golgi complex is present, it should bud off. This is lysosome. This vesicle will come and bind to this lysosome, releasing the protein inside the lysosome. So this is the process. So for this mannose binding protein, which is a lectin, is needed, and the recognition point is the mannose. Then only it can attach with the inner side of the bud or the vesicles, so that it can transport easily to the lysosome. So lectins, these are the proteins that read the sugar code. If you see the list of these lectin proteins, every protein is binding with a oligosaccharide or a carbohydrate, but with a specific carbohydrate. So which carbohydrate is present, depending on that particular lectin will come and bind and will mediate the corresponding function. Next is oligosaccharides also influence the circulatory half-life. What does it mean? Let's take an example of two hormones. One is luteinizing hormone and another one is thyrotropin. Now these hormones circulate in the blood. The function of thyrotropin is they release thyroid stimulating hormone. Function of luteinizing hormone is ovulation in female, as well as it helps in hormone production that needs for pregnancy. So once these hormones are released, they circulate in the blood. They'll circulate in the blood till it is needed for our body to function. Once that is done, the level of these hormones should get reduced from the blood. And how it will reduce? It will bind to a receptor present in hepatocytes. This receptor will recognize this hormone via the oligosaccharide present in it. Because of this specific oligosaccharide, this receptor will recognize and it will bind with this hormone. Once they bind, this interaction will mediate uptake and destruction of these two hormones. What will happen because of this? The concentration will go down in blood. So this how oligosaccharides also influence the circulatory half-life. That means for how long a particular hormone will circulate in the blood, that depends upon the oligosaccharide. So these luteinizing hormone and thyrotropin, they have N-linked oligosaccharides. That N-linked oligosaccharide end with this particular dimer. This dimer is recognized by a lectin, that is a receptor present in hepatocyte. Next is sialic acid. The other name of sialic acid is N-acetylnuraminic acid. This sialic acid functions as a protector of protein. But how? In the hepatocytes, we have a lectin receptor. The receptor is known as asialoglycoprotein. Asialo. That means without sialic acid. That means this receptor will bind to the protein which does not have sialic acid, and it will leave the protein which have sialic acid. So that means the proteins which lack sialic acid will bind to that receptor. So the receptor protein complex will form to the protein which lack sialic acid. Once this complex is formed, endocytosis will occur and the proteins are destructed. This is one mechanism by which the old proteins are meant for destructions. When the timeline or the lifetime of the protein is finished, or the function of the protein is finished, or the protein is damaged, or the protein is malfunctioned, in that case the sialic acid is removed from the protein so that the protein can be recognized by the receptor, which is asialoglycoprotein. That means it will bind to the glycoprotein which lacks sialic acid. After binding, endocytosis will occur and the proteins will get destructed. So this is one way by which proteins are marked for destructions. Next is selectins. These are the family of plasma membrane lectin only. From the name itself, you can see selectin. These are lectins only. They function in cell-cell recognition and adhesion. For example, in case of T lymphocytes, what happens during inflammation or at the site of infection? Normally, these T lymphocytes, they flow in our blood. Whenever there occur any inflammation or infection in the body, these lymphocytes from blood, it should enter into tissue. So how they can enter into tissue? The inflammatory site or the infection site, they will display P-selectin, where on the surface of capillary endothelial cell. This P-selectin will interact with specific oligosaccharide. And where this specific oligosaccharide is present? In the glycoprotein of T cells. So normally T cells flow in the blood, but whenever any infection occur or any inflammation occur, that time the T lymphocyte will enter from blood to the tissue. How it will enter? At the site of infection, there occur one lectin, that is P-selectin, which is displayed on the surface of capillary endothelial cell. This particular selectin will bind with a specific oligosaccharide which is present in the T lymphocyte, because of which the flow of T lymphocyte will slow down and it will adhere to endothelial cell. And after that, one more molecule comes into play, that is integrin. Because of integrin, then T cell can able to enter inside the infected tissue. So this is the function of selectin. Now, one question from my end for you all. Can you describe the mechanism used by Bordetella pertussis that cause whooping cough? Let me tell you one mechanism how Helicobacter pylori function. The Helicobacter pylori, they cause gastric ulcer. They have lectin that interact with oligosaccharide of glycoprotein of gastric epithelial cell. This is the mechanism how Helicobacter pylori can enter our body and can cause infection. Now you have to describe the mechanism by which Bordetella pertussis can cause infection, which results in whooping cough. Okay, do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.